right, today I'm going to present research that I did last year under Dr. James Albert on geometric morphometric comparison of Sternarchella sister species. Geometric morphometrics are used to study the shape traits within and among species. With these shape traits, we compare the differences between them and within them using multivariate statistics, which just means there's a bunch of variables that we are comparing and finding means, medians, modes, and p-values, etc., so on and so forth. Studying these shape traits or the morphology of these organisms helps researchers compare and map out the relations in trees in order to compile a full history of them. Phylogeny is important because it gives us a timeline of the world as these animals adapt and change. So, studying their morphology over time with the fossils that we've collected that have sat for years, and comparing them to current extant specimen that we collect tells us what kind of environments they've had to adapt to in order to survive in their time period. Understanding the traits that these animals have lost or gained, in this case morphologically, in order to survive in their environments, helps us to piece together their geographic features of the past and formulate conservation efforts for the future. So, why did we choose the Amazonian fish? What is the biological importance of this fish? Well, the Amazon is ancient and fruitful. It's an environment that has a high level of species richness. The river itself harbors different habitats and niches at its different trophic levels. And while there aren't that many complete fossils from this area, because of the constant change of the land and the geography around it, many of the fish that we've collected or that others have collected show relations to one another, yet live on opposite sides of geographic barriers. The Amazonian fish evolved pretty quickly, and quickly doesn't mean within our lifespan, but in evolutionary history, 5,000 years can seem like the blink of an eye. And these fish happen to evolve in about that time period. They can be the key to understanding how the ecology and geography and the change in those things contribute to adaptation, even in a short span of time. So the Amazon River Basin is obviously a river basin, and it's vast and encompasses a large amount of South America. The river itself, the large river, the very famous Amazon River, has an ever-changing direction of flow. So over a few thousand years, the Amazon will change direction and create the quickest path to its destination. And while it's doing this, in its wake, it leaves behind what we call tributaries, which are really just smaller rivers that feed into the Amazon. This also creates new watershed divides and causes the geographic change we mentioned before. Over time, the flora and the fauna, like the fish and the animals of the Amazon, have to adjust their own physiology to adapt and survive in this environment. So as this river changes and separates these original populations from themselves and within themselves, this allows them to speciate and diverge from their ancestors, which brings us to the topic of sister species. A sister species is two or more species that have the same immediate ancestor, but speciated because of geographic barriers. And again, geographic barriers could be the rising of a mountain or the movement of a river changing, or maybe even, lava erupted and an island was completely revamped. So this kind of speciation is actually called allopatric speciation. Allopatric just meaning a geographic separation, causing them to speciate, as compared to sympatric, which is speciation driven by other forces. So here on the screen, on the left is Sternarchella calamazon, and on the right is Sternarchella patriciae, and these fish look almost exactly the same. The differences between them are very small and they're morphological. These are actually a couple of sister species we're going to study in this study, but this here is just an example of a very closely related pair of sisters. Here I have an example of one of the visual aids that researchers use to get across the point for the relation of one species to another. Each branch here is a lineage, and each end is a species. So if you look closely, you can see Sternarchella sima and Sternarchella orinoco here. These two species are sister species, and we know this just from looking at this by seeing that their branches converge into one common branch, which means they share an immediate common ancestor. And that's an example of a sister species. So the specimen we actually used for this research are on the right, these pink little naked guys. They are from Gymnotiformes apteronodidae Sternarchella species, and they are from one of the oldest families that have deep adapted to their environment by losing traits they no longer need. The Amazon River is so deep and dark and muddy that these fish no longer need color variation or sight to navigate the world around them. So those traits are lost, which explains why they appear to be naked. So the way that they actually navigate and communicate with each other and with the world is through electric organ discharge. And what's very interesting about these fish is that each species, has its own unique electric organ discharge that can be measured and matched to them. So that's just an interesting little fun fact about these freshwater electric fish. The sister species that we actually used from this collection were Sternarchella calamazon, Sternarchella patriciae, and Sternarchella sima, Sternarchella orinoco. And they are sister species to one another. These were collected in the upper and lower basin and are very abundant in the Amazon River basin area. They also happen to be very abundant in our lab at the time. We probably had over 200 fish just from the Amazon, and then about 50 of those were probably just patriciae, honestly. So what we did is we chose at least 10. Some of them had a few more because some tails seemed kind of like outliers. So we chose at least 10 of each of these specimens to be cataloged and counted. And these were all collected by a team of ichthyologists in the late 90s. So for each fish, we marked 17 landmarks on their bodies with insect pins. And we knew where these landmarks were because researchers before, have been able to pinpoint the important morphological landmarks on fish of this kind and provide charts and data and on this type of landmarking. And they've given us these kinds of models. So where every red dot is on these two model fish, the cartoon fish here, is where we've put an insect pin to mark those unique morphological traits. We then place these fish on a gridded background, as you can see in the upper right-hand corner, this fish, and then photograph them with high-definition cameras. We only use the fish that appear to be full length and fully grown. And that's really important because in the wild, these fish actually get their tails bitten off quite a bit. So a fish that can appear to be a juvenile fish might actually be a fish that's just in the process of regenerating their tails. So we had to also do the research and figure out the appropriate length for an adult fish because depth is not something that diverges from their juvenile states. So the best way to tell them apart is looking at their length. The photos that we took of both the head and full body were then uploaded to this software called ImageJ, and then pixelated, and then turned into an XY coordinate plane. And from this is where we got our quantitative data. As you can see, these bright green dots outline the XY coordinates that would soon be put into a chart. It's just a good visualization of what happened on ImageJ. From ImageJ, we uploaded these XY coordinates into Morphometric, which is just another shape analyses software that researchers use to do geometric morphometrics. And then with this newfound quantitative data, we were able to find the mean points, which if you look at the picture on the left, these larger blue dots are average points, and there's a bunch of smaller little dots around it that quantify the landmarks we've marked for all of the other fish. The first thing we did with this information is a Procrustes fit, which is just a shape analysis used to analyze the distribution of shape in these fish. As you can see, this set of data on the left superimposes directly on top of this fish, with the average points almost lining up exactly on top of the pin landmarks. We then performed what is called a principal component analysis, which just analyzes each of these, we're going to call them dots, each of these dots or data points as components and compares them to one another. And the model that we get from this is called a ball and stick model. And this is just an example of one of our resulting ball and stick models of a head measurement. So as you can see on 13 and 12, there's a ball and a stick. And what the stick signifies is how much variance their data had in that set. So 13 and 12, there's a lot of variance in the set that we entered for those landmarks. And then if you look at 1 and 2, I'm not sure if you can see them, but it's pretty much the snout and right below where the eye would be. 1 and 2 don't have as much variance as the others. And this is just like a way to visualize where the highest variance might be with these models. So for Sema and Orinoco, our first pair of sister species, we had principal component 1 and principal component 2 show the most variance. So the first principal component is actually based on body depth. So I have here the red circles around where you can see the variance for body depth anteriorly and posteriorly. Those points are 13, 14, and 17, which if you were to look back, you can see that those are some of the morphologically important landmarks on these fish. The other second greatest amount of variance is in principal component 2, which is a variance in length for Sema and Orinoco. And again, the ball and stick model shows that there is a large amount of variance here. But this is only a visual model, and it's not many numbers that can come from this unless you plot it against each other. So here I have a scree plot, which is just a plot of present variance against the principal components. See, the principal components, there are 10 or more because there's 10 or more landmarks that we actually analyzed. And the first two show the highest variance. That's principal component 1 and principal component 2, being depth and length, respectively. Following that, we plotted the actual principal component analyses against one another to determine their orientation to the axis of greatest variation, which is a mouthful, but really it's just the variation between these two species. And that's represented by this dashed red line going straight through the middle here. And the Pmax and the Gmax represent the variance within the species, how the species vary within their own personal families. As you can see, the variances within the species are not parallel to the variances between the species, which means that the forces that shape the patterns of covariance in the species is not the same as the forces that shape the variance between the species. We ran the same test on Patriciae and Kalamazon's data, and we got similar results, except there is less significance for the variance in length. More of the significance lied in the variance for depth, and that just is just very significant morphological difference for both pairs of sister species. That's what we concluded from that information. And this is just another scree plot. You can see here the highest amount of variance is principal component 1, which is depth, with length trailing rather far behind as a principal component. So again, that just is very significant. Depth is very significant for identifying one species from another for both pair of sister species. When we then plotted these principal components of these sisters against one another, they also suggest the same as Sima and Orinoco. These results from this principal component and the one before are actually consistent with the interpretation that sister species pairs of Sternarchella, the fish in this study, evolved in allopatry across the Amazon-Orinoco watershed divide, but under similar ecological conditions, which means they had the same types of habitats and the same types of dietary conditions, which means that the only force really driving the divergence from their ancestors would be the forces of geographical change. I'm sorry. So the information on the morphology of these closely related species, the sister species, gives us a lot of evidence on the effect of ever-changing environments and the effect it has on morphology and adaptation of populations. So the separation of just two watersheds over a few thousand years can cause one population to diverge into two completely different species. And it provides information that will also allow researchers to form conservation efforts in the future, because we can see how quickly these fish can adapt and how much time they need, and what environment would be a successful environment for them. So this is really effective for helping us formulate conservation efforts in the age of technology and habitat destruction, in the Anthropocene, if you will, the age of the humans. So that's the biogeographic importance of this study. Any questions?